Hi guys, how are you all? I'm pretty sure it must be so amazing, and we are just going to discuss our part 2, excrete product and their elimination. I'm pretty sure you have seen part 1. Write down in the comment box if you have seen it or not, and I'm pretty sure you like the video. And which are today, so yesterday we are done with the structure of the kidney. We are done with the structure of nephron. We have seen all the part, we have seen all the function. Now we will be starting physiology, that is the urine formation, and we will be seeing disorder today, right? So are you ready? And bacha, we will be doing like all the videos in one shot or two shot now, right? So let's start it now. Excretory product and their eliminations. So we are going to start with the urine formation. So we know that urine formation will be happening in occur in three steps. One is the glomerular filtration. Glomerular filtration is very simple. That blood will come from afferent arteriole, you know it very well. And obviously it will enter the glomerulus, which is the tuft of capillaries, which will filter the blood out in the Bowman's capsule. So this is the Bowman's capsule. It will filter the blood here. Bowman's capsule mein blood aayega, filtered blood, urine, right? And then obviously it will go ahead. So we will be seeing this is the ultrafiltration, glomerular filtration, and after filtering blood will exit in the efferent arteriole, that we know, right? We have already done. And thanks a lot for your huge love and support. Those students who want notes and any doubt, they can send me there in the Telegram channel. I've given you link below in the description box of this video. You can check it out. Right, so bacha, yahan par obviously blood is coming, filtering out from the glomerulus, and then blood after filtering exiting out in the efferent arteriole that we have seen yesterday also. Reabsorption means there are obviously when the filtration happened, when your mother asked you to clean your room. So what you have done, you have you have just put all the useful and not useful things, and you put all the things outside. So now that is the glomerular filtration, which is non-selective process, means it is not selective. It is not selectively filtering things. Useful things also filtered out. So what we have to do now, all the useful things need to be sent back in the blood. And we know efferent arteriole branches known as peritubular capillaries. So obviously those reabsorbed thing will again go back to the efferent arteriole, in the peritubular capillaries. That is known as reabsorption. So useful things will be going back to the peritubular capillaries, and sometime blood up yahan par, that is to reabsorption means tube say obviously renal tube is actually sending back useful things to the blood. That is the reabsorption. तो ट्यूब से चीजें जा रही हैं वापस ब्लड में. That is reabsorption. And secretion means still blood have some unuseful thing, right? तो अभी भी blood के पास कुछ unuseful thing हैं. There are unuseful thing in the blood, so obviously it will be coming back to the renal tube. So blood से कुछ unuseful चीजें या फिर कुछ waste material still has to come to the tube. That is known as secretion, and we will be doing all the things in detail, right? So bacha, are you ready? So these are the three steps that we are going to elaborate, and let's start with the glomerular filtration. So we know that glomerular capillaries have high blood pressure. Why? Because I have told you efferent arteriole ka diameter is broader. So it is having the broad diameter, and obviously sending the blood in the glomerulus with high flow blood is there. Whereas efferent arteriole is the narrow. So obviously narrow means less blood will be going out, right? So that means when more blood coming out, less blood going out, it will create high pressure here. And because of this high pressure, things will filtered out, even useful thing also filtered out in Bowman's capsule space. We know it very well. So bacha, yes, glomerular capillaries blood pressure cause filtration of the blood through three layers. So obviously we are having three layers for the filtration. What are those? One is the endothelium of the glomerular blood vessels. So we know that glomerulus का जो capillaries है, capillaries का जो wall है is made up of the endothelium. So we are having the endothelium layer of the blood vessel, that is the glomerulus. Then obviously endothelium means it is the squamous epithelium, endodermis in origin का squamous epithelium है forming the glomerulus blood vessel. This is the basement membrane, and we know that basement membrane is acellular structure. Basement membrane is acellular structure, not made up of any cell. and it provide base to where the endothelium rest and where the squamous epithelium of the Bowman's capsule also rest. So Bowman's capsule का जो inner layer है, I have told you it is the modified squamous epithelium podocyte cell. So they are having the feet-like cell, podocyte cell, and these podocyte cell have the slit in between. So slit pore is there, slit pore opening is there, right? So slit pore filtration slit, sorry, it is known as pore or filtration slit. So it is known as filtration slit. So things from the blood will filtered out and come here in the Bowman's capsule. So this is the Bowman's capsule space. This is the capsular space. So things are basically collected here. So obviously filtration membrane made up of 3 layers. One is the endothelium of glomerulus blood vessel, squamous epithelium, or we called it modified squamous epithelium of the Bowman's capsule known as podocyte, have the filtration slit, filtration pore, through which the things will filtered out from the blood and come here. Here the glomerular filtrate will be collected, right? And basement membrane is there, which is acellular layer. Total 3 layers are there, which help in the ultrafiltration, means fine filtration of the blood, right? So first step is the ultrafiltration. So we know that ultrafiltration is the fine filtration of blood, fine filtration of blood. And obviously when this blood filtration has happened, what will happen next? Filtrate will form. So we know that obviously blood has come from the glomerulus and it just enter the capsular space. This is the Bowman's capsule space, right? So this is known as ultrafiltration, and whatever filtered out known as glomerular filtrate. Glomerular filtrate. So this is the first step of the urine formation, that is the ultrafiltration, as simple as that. I have told you, due to high pressure, useful plus non-useful things all are basically moved here, right? All are basically filtered here. So ultrafiltration is a non-selective process. Non-selective means useful plus non-useful things all are basically come out, right? So this is the non-selective process which you have to remember. During filtration, what happened? All the constituent of plasma actually filtered out, right? So you can say blood is finally filtered. All plasma constituent, means almost composition of the plasma constituent, filtered out in the space, in this Bowman's capsule space, filtered out except protein. So obviously plasma में से you just minus protein, whatever there is the plasma, just you remove the plasma protein, and whatever is there will filtered out here, right? So that is actually the glomerular filtration, ultrafiltration, because of high pressure blood is finally filtered out. That you have to remember. तो मतलब इसमें plasma का composition without protein है. So that you have to remember. तो कितना filtrate बनेगा kidney से per minute? So amount of the filtrate formed by kidney per minute is known as glomerular filtration rate, and it will be 125 ml per minute, right? And if we see per day, so it will be 180 liter per day. So per day, obviously this much glomerular filtration rate. This is the GFR. This is the GFR, glomerular filtration rate per day. तो 180 लीटर टोटल छनकर आता है पूरे दिन में. तो blood is coming several times in the kidney, and this much filtrate per day will be filtered out. But GFR is controlled by juxtaglomerular apparatus. What is it? I'll be telling you, right? And obviously 99 out of the 180 liter per day filtrate, 99% वापस से reabsorb हो जाएगा. So whatever has come filtered out, obviously 99% again go back to the blood. That is known as reabsorption. So second step we will be seeing reabsorption, right? And remember, ultrafiltration is basically non-selective process, means useful, non-useful, all the things has come filtered out. But now non-useful thing will again, useful thing again go back to the blood, right? So we will be seeing reabsorption. Bacha, it can be active. Active means it requires energy, or it can be passive. So some element will be reabsorbed actively, like sodium ion, like all sugar, like all amino acid. These all will be actively absorbed. Passively, like water via osmosis, will be absorbed, reabsorbed back, right? So obviously nitrogenous waste also. So this is something which you have to remember will be passively, means other electrolytes. So nitrogenous waste to secrete hoga. So if we see, yes, so electrolyte also will be reabsorbed. Some electrolytes like sodium ion, sorry, like chloride ion and all, you know. So chloride ion or this potassium ion and all will be reabsorbed back in the blood with the help of this, passive transport, right? So that you have to remember, not require energy. And I have told you 99% filtrate, glomerular filtrate will be reabsorbed. Will be reabsorbed. As simple as that. And obviously you have to remember that PCT, when we talk about reabsorption, it is the selective process, means selective things will be reabsorbed back. So this is something which you have to remember. Plus when we talk about reabsorption, the maximum reabsorption happen in the PCT. I have told you why, because it is lined with the brush border cuboidal epithelium in the previous class, right? So PCT may maximum reabsorption will happen. And the another one is, out of one, and minimum reabsorption कहाँ पर होता है? तो minimum reabsorption happen in the ascending limb of reabsorption. अगर हम पूछें कि कहाँ होता है, तो it will be happening in the ascending limb of loop of Henle. So loop of Henle के ascending limb में, that you have to remember, right? So this is all the thing about the reabsorption you have to remember. And reabsorption may, you know what, once the thing once the things get reabsorbed back, only out of 180 liter जो filter होकर आया था, out of 180 liter that were filtered out per day, only one out of that, only 1.5 liter urine are produced, are excreted out, are excrete out per day, or are released out per day out of 180 liter glomerular filtrate per day. So obviously इतना ज़्यादा glomerular filtrate बनता है, but out of that 90% get reabsorbed back in the body, है ना, glomerular filtrate. So that's all. This is all about the reabsorption. So बच्चा, reabsorption is very important here, and you have to remember that where this happened. And the third step is the tubular secretion. So reabsorption is done. Again, I have told you some harmful things still remain in the blood. So from the blood, thing will come to the tube again and become the part of urine, and that we will be seeing. So secretion is always active. तो मतलब secretion किसी भी चीज का हो रहा है, हमेशा याद रखना, it will be active. With help of energy will happen. It is also selective process. Selected things will be reabsorbed, right? So will be secreted, sorry. So this is the selective process. And when we talk about tubular secretion, why it is required? So obviously tubular secretion is required, help in maintaining pH, acid-base balance, So this is the purpose of the, this is the purpose of this, what we called secretion, tubular secretion, even ionic balance, ionic balance of body fluid. So obviously our body fluid balance, acid-base balance, ionic balance, pH maintained by the secretion only. So what are the things that is secreted out from the blood to the tube again? So secretion of the hydrogen ion, that will be maintaining acid-base balance obviously, potassium ion, even some nitrogenous waste, Hyperic acid, this is something which you have to remember. Hyperic waste, so Hyperic acid is basically metabolic waste from the benzoic acid. So it is produced from benzoic acid. So you know what is benzoic acid, तो ये वहाँ से बनता है, right? So it is formed from the benzoic acid, you have to remember. Another one is the creatinine. So when we talk about creatinine, it is produced from creatinine phosphate of muscle. Creatinine phosphate in muscle. तो मतलब ये muscle से generated एक waste है, which will be secreted from the blood back to the tubule, right? उसके अलावा drugs हो गया, drugs will be secreted out, toxins will be secreted out from the blood. Even drugs, pigments will be secreted out from the blood. And you know what, although we are not so much depend on secretion, because most of the things filtered out from the ultrafiltration, from the glomerular filtration, but यहाँ पर secretion is so useful for those animals who don't have glomerulus, right, who don't have the capillaries. So glomerulus capillaries, if absent in those animals, so what will happen? Ultrafiltration will not happen. So they totally depend on the excretion. They totally depend for the filtration, or they totally depend, if they want to excrete something in the urine, they totally depend on the excretion. So what are the examples? Those who don't have the glomerulus, तो बच्चा desert amphibians हो गए. Marine fishes, they don't have like well developed kidneys, so they don't have glomerulus well developed, so they totally depend on secretion for the excretion, right? So that you have to remember. So now we will be seeing the function of tubule. तो यहाँ पर एक एक tubule कैसे कैसे काम करता है. तो we have seen Malpighian tubule, Malpighian corpuscle, that is glomerulus and the Bowman's capsule help in the ultrafiltration. So second, we will be seeing the PCT, proximal convoluted tubule. We know maximum reabsorption happen here. And मैंने कहा कि obviously this reabsorption happen in the various tube. So suppose this is the PCT. And when we see the PCT, so obviously this is the fluid outside the proximal convoluted tubule. So obviously PCT is the isotonic. Isotonic means it is having the same osmolarity as outside. So this is the isotonic, then it is the isotonic, it is containing isotonic filtrate as outside, right? So means यहाँ पर जो है reabsorption किस चीज का होगा, and you have to remember किस चीज का secretion होगा इस tube से. Secretion means what thing from the blood go to the PCT, and reabsorption means from what piece, from what substance from the PCT come to the blood again, right? So we will be seeing it. So बच्चा reabsorption 80%, 70 to 80% electrolytes. Electrolytes means obviously ions. Sodium ion actively with help of energy. Rest इसके पीछे पीछे जाएंगे, जैसे chloride ion will just move passively along with the sodium ion. Bicarbonate हो गया, potassium हो गया, so they will be moving passively again in the blood, right? 70 to 80% मतलब maximum water reabsorbed here by osmosis, passive process. And remember bacha, they will be, this tube will be showing the obligatory reabsorption of the water. Obligatory means without any condition, maximum water will be reabsorbed, maximum thing will be reabsorbed here. So obviously it is the obligatory, मतलब होना ही होना है, there is no condition at all. It will happen, obligatory reabsorption will happen. Then obviously all glucose, all sugar, all amino acid will be reabsorbed, and obviously both of this will be reabsorbed actively. So this is the active process with help of ATP will happen. What is secreted? So secreted means obviously this blood from the blood, things are coming to the tube again. तो किस किस चीज का secretion होगा? Obviously hydrogen ion. I have told you they are active process always. This is ammonium ion, right? So not potassium, but yes, hydrogen ion. So hydrogen ion, sorry. So we are having this hydrogen ion, this ammonium ion, we are having toxins, we are having pigments. So these are coming back to the body, pigment, even hippuric acid. You know what is hippuric acid? I have just told you, right? Hippuric acid सब वापस आ रहा है from blood to the tube, creatinine. So all the bad thing from the blood again come to the part of the, again become the part of urine, part of filtrate, right? So this is all about the PCT, क्या reabsorption होगा, क्या secretion होगा, कैसे. This is important for exam. Now the second part is the after PCT fluid will go to the loop of Henle, right? So the second part will be loop of Henle, and loop of Henle have significant role, and this is very important point. Significant role किसमें होता है? Significant role in maintaining high osmolarity in medulla. And you know what is medulla, right? Maintaining very high ऑस्मोलैरिटी मीन्स सॉल्यूट बढ़ाता है बहुत ज्यादा इन मेड्यूलरी इंटरस्टिशियल फ्लूइड. You know medulla region. So whatever space is there, whatever medulla may space filled with the fluid will be having the more osmolarity, which is maintained by loop of Henle, right? So medullary fluid osmolarity, or you can say interstitial medullary fluid osmolarity will be higher. So this is our, obviously this is our kidney have the medulla, this is having nephron. So this space obviously is filled with the medullary fluid. So बच्चा ये medullary fluid में it will maintain high osmolarity, and why we will be seeing it in detail way. High osmolarity, you know, means high concentration of solute, and we know loop of Henle's are of two types. What are those? Ascending and descending limb. So let me just tell you what is going to be reabsorbed in ascending limb and what is going to be reabsorbed in descending limb. So one is the descending limb, another one is ascending limb. So when we talk about descending limb, no, याद रखना descending limb also known as concentrating limb, because I have told you this limb is water permeable, right? And when it is water permeable, क्या होगा इस कंडीशन में? यहां पर अंदर बन जाएगा hypertonic condition. I'm pretty sure you know what is hypertonic. Highly concentrated. क्यों? क्योंकि it is water permeable. Water will be reabsorbed in the medullary fluid, and water के बाहर निकलने से अंदर concentration solute का बढ़ गया, and hence water कम हो गया, and it become concentrating limb now, right? So means it becomes so much highly concentrated, it is reabsorbing water. And if we see the secretion, no secretion, nothing secreted here, only reabsorption of water, because it is water permeable and obviously forming the hypertonic solution inside, highly concentrated solution inside. And when we talk about ascending limb, तो बच्चा simple, ascending limb have two parts, thin part, thin ascending limb, and thick ascending limb. So remember, thin ascending limb reabsorb all the things passively, passive reabsorption. Whereas in case of the thick limb of ascending limb, active reabsorption happen. So we will be seeing both the things, and obviously seeing what is reabsorbed here, what is secreted here. Reabsorption or secretion किस चीज का है? And ascending limb is known as diluting limb, because it is having the diluted solution. Why? Because we know that it is solute permeable but water impermeable. So obviously जो solute इसके अंदर होगा hypotonic होगा. Hypotonic means having the low salt content. Hypo means low, tonic means concentration. Low concentration means इसमें salt कम है और पानी बढ़ गया. Salt क्यों कम है? Because NaCl comes out, right? तो NaCl comes out passively, we know it very well, and some amount of little amount of urea secreted in it. Why? We will be seeing it in the counter current mechanism. Little amount of urea secreted in the thin limb of ascending limb, right? Thin limb of the loop of Henle, that you have to remember. Active इसमें अगर हम देखेंगे obviously, तो here NaCl is reabsorbed actively. तो here only reabsorption happen, no secretion. कैसा होगा? Hypotonic condition in ascending limb. Why? Because solute releasing out and पानी का content अंदर बढ़ रहा है, making it less concentrated. As simple as that. So this is all about ascending limb. And बच्चा, in case you find any doubt or if you find any difficulties understanding these process, feel free to send me doubt in the Telegram channel, given the link below in the description box. So please check it once, right? So now finally, and you have to remember that obviously as the fluid pass upward in the ascending limb, so obviously it becomes so diluted. So that's why ascending limb is known as diluting limb, because limb के अंदर पानी ज़्यादा है, salt कम है, it becomes so much diluted. Again, very important part, right? When we talk about distal convoluted tubule and collecting duct, so बच्चा distal convoluted tubule and collecting duct में जो reabsorption है, it is conditional. It is conditional only in certain condition, or we call it facultative reabsorption. कंपल्सरी नहीं, जब कंडीशन होती है बनेगी तभी रीअब्जॉर्प्शन होगा, ओनली ड्यूरिंग सम कंडीशंस, राइट? So conditional or facultative reabsorption happen when there is the stress of water and the salt in the body, then only it will get activated. So obviously it is basically conditionally activated by ADH, that is antidiuretic hormone. And we know antidiuretic hormone is released from posterior pituitary gland. As simple as that, posterior pituitary gland. And what is its purpose? Obviously it will be helping in the water reabsorption. We will be seeing it. It will be helping in the water reabsorption. And second hormone is the aldosterone, and obviously it is secreted by adrenal cortex gland, right? And what is its purpose? Sodium balance. So it will be reabsorbing sodium. It will help in sodium reabsorption from DCT. So we will be seeing it in some condition, right? So obviously when we see DCT, we will be seeing what is reabsorbed here, what is secreted here. तो बच्चा DCT में क्या क्या reabsorption होगा? Conditionally हम देखेंगे, इसमें बाइकार्बोनेट रीअब्सॉर्ब होगा, कंडीशनली वाटर व्हेन देयर इज द ADH प्रेशर, ADH का कंडीशन है, एंड ऑब्वियसली इट विल बी रीअब्सॉर्बिंग सोडियम आयन ओनली व्हेन देयर इज द एल्डोस्टेरोन. तो मींस इट इज द कंडीशनल, ऑब्वियसली इट्स कंडीशनल, एज सिंपल एज दैट. एक्टिवली सोडियम आएगा, लेकिन बच्चा ऑब्वियसली इट इज ओनली कंडीशनल. सो मैक्सिमम सोडियम रीअब्सॉर्ब्ड इन PCT एक्टिवली, यू हैव टू रिमेंबर, राइट? So when we talk about secretion, तो वही hydrogen ion secreted from the blood back here, potassium ion, ammonium ion, just to maintain ionic balance, acid-base balance, right? Same when we talk about collecting duct, it will be releasing water, it will be reabsorbing water only in the presence of ADH. When there is the requirement, it will be absorbing water, right? And obviously little amount of urea also reabsorbed. Why little amount of urea is again sent back here in the medullary fluid? Just because medulla के space में urea आएगा to maintain high concentration, to maintain high osmolarity in medullary fluid. तो यहाँ पर हमारा पर्पस ये है कि मेड्यूलरी फ्लूइड में हाई कॉन्सेंट्रेशन बढ़ा के रखनी है. क्यों? आई विल बी टेलिंग यू. एंड सिक्रीशन किस चीज का होगा? तो फ्रॉम ब्लड हाइड्रोजन आयन, पोटेशियम आयन, अमोनियम आयन, टू मेंटेन द आयनिक बैलेंस, टू मेंटेन एसिड बेस बैलेंस will be done here in the collecting duct. As simple as that. I have just given you very simplified explanation here, and the next step we are doing the concentrated urine formation, which is again very important, and this is NCERT diagram. तो बच्चा see this diagram very nicely. तो obviously this is the PCT, है ना? देखो यहाँ पर this is PCT, and PCT में all this are reabsorbing back in the blood, and these are secreting back in the urine form. Water, descending limb is water permeable, water coming in the medullary fluid. This is the fluid. And obviously ascending limb releasing NaCl because it is the solute like permeable. So obviously it is releasing medulla में NaCl. Then this is the DCT reabsorbing all these things back, and this is secreting blood से वापस चीजें यहाँ पर आ रही हैं. And this is the collecting duct. So obviously it will be releasing some amount of water, reabsorption of water, and small amount of urea also in the medullary space, in the medullary fluid. And obviously time to time this urea will be secreted in the ascending limb, that I have told you in the previous session also, previous slides also. And ये urea ऐसे cycle करता रहता है. तो urea is secreted here in the ascending limb, then again coming to the collecting duct, again going out. And bacha, we are not complicating any process, because I'm telling you only those things, the simplest way that they will definitely ask in the exam. right? And you will definitely be able to solve question from any of the modules. So just focus here, right? So बच्चा medulla में obviously high osmolarity हमें maintain करनी है. We have to maintain high osmolarity in this medullary space, and which is done by the loop of Henle, as I have told you. Why we have to maintain high osmolarity here, I will be telling you. Even urea also help in maintaining high osmolarity in medulla. How, why, we will be doing it, right? So before going ahead, there is a question for you, that maximum amount of electrolyte and water, 70 to 80% from the glomerular filtrate, is reabsorbed in which part? Easiest one. I have already mentioned the amount, and this is the NEET 2012 question, right? There is the another diagram they have given, beautiful diagram, which is basically showing the counter current mechanism. What is counter current mechanism? Let me just tell you. So bacha, remember that obviously this is the glomerulus filtering thing, and some things are going back, some things are secreted. Now we will be seeing the concentrated urine कैसे बनता है, and let me just tell you how to form the concentrated urine. So counter current mechanism का जो purpose है to form the concentrated urine. So when we want to form the concentrated urine, we will be doing counter current mechanism. Why do we call it counter current mechanism? तो we know this is the loop of Henle, ये loop of Henle है, and just parallel to loop of Henle, vasa recta means blood vessel is there. लेकिन NCERT ने क्या करा, vasa recta को they have just put in one side, avoid confusion, right? To avoid confusion, and this is loop of Henle. So simple सी बात है यार, descending limb of loop of Henle. Now see this diagram very carefully. As the solute coming down, this is the descending limb of loop of Henle. It is basically, filtrate going down this way, in this direction. And ascending limb में filtrate going upward this way. You can see in the diagram. So filtrate का जो direction है, it is opposite. One descending limb में filtrate coming down, and ascending limb में filtrate coming up, going up, right? So this is basically the counter current. Counter current means obviously filtrate moving in the opposite direction. As simple as that. When we talk about blood का flow, तो we know efferent artery. This is efferent arteriole की branch होती है, peritubular capillaries and vasa recta. So this way present around it. So blood after filtration coming downward in this tube. So blood is coming in this direction here. So इस वाली tube में it is coming. This is the descending limb of vasa recta. So blood coming down in here, and obviously blood going up in the ascending limb in this limb, right? So means descending limb of loop of Henle is just present parallel to the ascending limb of vasa recta, and ascending limb of loop of Henle is just parallel to the vasa recta का, like descending limb of vasa recta. तो यहाँ पर ब्लड नीचे की तरफ आ रहा है, फिल्ट्रेट ऊपर की तरफ जा रहा है, तो दे आर ऑल्सो हैविंग काउंटर करंट, अपोजिट डायरेक्शन मूवमेंट. एंड हियर ऑल्सो ये फिल्ट्रेट नीचे आ रहा है और ये ब्लड ऊपर की तरफ जा रहा है. दे ऑल्सो ब्लड गोइंग अपवर्ड, फिल्ट्रेट कमिंग डाउनवर्ड. दे ऑल्सो हैव द अपोजिट अरेंजमेंट. सो ऑल द 4 ट्यूब्स हैव अपोजिट अरेंजमेंट ऑफ देयर secretion, or arrangement opposite, this fluid moving in the opposite direction in all the 4 tubes, right? So that means counter current mechanism, concentrated urine का formation will be done with the help of the loop of Henle and vasa recta. Loop of Henle and vasa recta have huge role in maintaining concentrated urine. So if we want to produce concentrated urine, these 2 tubes will be helping. This blood ka supply will be helping, and this filtrate supply in the loop of Henle helping. And both of them are arranged in counter current fashion, means opposite current इसमें जा रहा है, opposite flow जा रहा है, right? Arranged in counter current fashion. Counter current fashion means opposite fashion. As simple as that. So bacha, now we will be seeing that how this will help in the counter current mechanism. Now listen to me very carefully. Whatever I have taught you so far, right? Whatever I have told you so far in the previous classes, previous lectures. So now you see. यहां पर क्या होगा कि obviously filtrate coming in the PCT, many things reabsorb secreted, we know it very well, and descending limb releasing water outside, and obviously because water बाहर जा रहा है, बाहर की तरफ मेड्यूला में जा रहा है, तो मेड्यूला में जैसे जैसे पानी जा रहा है तो ऑब्वियसली डिसेंडिंग लिम्ब बिकम हाईली कॉन्सेंट्रेटेड, इसमें सॉल्यूट बढ़ रहा है, सॉल्यूट इंक्रीजिंग इनसाइड इट एंड इट बिकम हाईली कॉन्सेंट्रेटेड, हेंस डिसेंडिंग लिम्ब इज कॉन्सेंट्रेटिंग लिम्ब. Up now in the ascending limb, water is coming, filtrate is coming upward, going upward, and NaCl releasing out because it is permeable for solute. So obviously it is decreasing salt inside and increasing water inside. तो water बढ़ रहा है तो मतलब it is become diluting now. और कितना dilute हो गया? 200. तो बच्चा this is the way I have told you that descending limb is concentrating limb and ascending limb of loop of Henle is diluting limb, right? तो हमारा जो पर्पस है ना, what is our purpose? This is our cortex region of kidney and this is the inner medulla region of kidney nephron. You know it very well, right? Cortex region की osmolarity 300 है, 300 which is like 300 milli osmolarity हमारी maintain होनी चाहिए, right? So our body fluid have this osmolarity, our blood also have maintained 300 milli osmolarity. इतना blood में होना चाहिए. This need to be maintained. But deep medulla have the 1200 milliosmolarity, which is very high, means it is having various solute in it. पानी कम है, solute ज्यादा है, तो means cortex and medulla region, तो मतलब यहाँ पर cortex have 300, it is having 1200 milliosmolarity, means 4 times more. So osmolarity here 4 times more than the cortex region, as simple as that. So बच्चा osmolarity कैसी है? Osmolarity means concentration of water and solute. So obviously if we see cortex से medulla region में it is 4 times higher. And you know what? Why we have to maintain 4 times more osmolarity in medulla? I will be telling you. So our purpose is to maintain high osmolarity in medulla, high concentration in medulla, high salt in medulla, right? हाई ऑस्मोलारिटी मेंटेन करना हमारा पर्पस है. दिस इज आवर पर्पस. हाई ऑस्मोलारिटी इन मेड्यूला, इन मेड्यूलरी फ्लूइड. व्हाट इज व्हाई? आई विल बी टेलिंग यू. So when there is the high osmolarity, what will happen now? See, listen to me very carefully. How it will be maintained? How high osmolarity will be maintained by the vasa recta and loop of Henle. तो बच्चा now see it very carefully. हाई ऑस्मोलैरिटी ऑफ मेड्यूला कैसे मेंटेन होएगा? तो इसके पीछे 2 रीजन है. 2 ऑस्मोलाइट इज देयर. हाई ऑस्मोलैरिटी इन मेड्यूला मेंटेन्ड बाय, सो ऑब्वियसली हाई ऑस्मोलैरिटी हमें मेंटेन करना है. कितना? 1200, विच इज वेरी हाई. हाई ऑस्मोलैरिटी ऑफ मेड्यूला मेंटेन्ड बाय, एंड हमें क्यों मेंटेन करना है? आई विल बी टेलिंग यू. बिकॉज वी हैव टू प्रोड्यूस वेरी कॉन्सेंट्रेटेड यूरिन एज वाटर स्ट्रेस इज देयर इन अवर बॉडी. सपोज बहुत गर्मी है, स्वेटिंग इज हैपनिंग, वाटर लॉस हैपनिंग फ्रॉम द बॉडी, सो वी हैव टू कंजर्व वाटर via मतलब conserve water and we have to release concentrated urine. So high osmolarity of medulla maintained by NaCl and urea. Bacha, this is the neat question. NaCl, urea, these are 2 osmolites which will maintain high osmolarity, high concentration in the medulla. कैसे? Now see it. देखो, obviously ascending limb, if we see the ascending limb, now see this flow chart. Ascending limb releasing NaCl here, and this NaCl will be maintained by blood that is coming down from the vasa recta. Blood जो आ रहा है नीचे, it will be maintaining this NaCl outside, मतलब it will be absorbed by the NaCl present outside. तो देखो यहाँ पर ये डायग्राम, NaCl जो बाहर आ रहा है, तो NaCl which is releasing from the ascending limb is absorbed by the vasa recta just to maintain osmolarity, and it is releasing water outside, blood releasing water outside. तो बच्चा इसके वजह से जो ब्लड आया नीचे जाते जाते ब्लड बिकम हाईली कॉन्सेंट्रेटेड बिकॉज इट इज हैविंग मोर सॉल्यूट एंड लेस वाटर. बट एज इट इज मूविंग अपवर्ड, एज ब्लड मूविंग अपवर्ड, तो बच्चा वी नो दैट वाटर कमिंग आउट एंड दिस वाटर को मेंटेन करने के लिए ह्यूज NaCl विल बी रिलीज्ड बाय द वासा रेक्टा आउटसाइड, and water will be absorbed, some water will be absorbed by this ascending limb of vasa recta from here. So that means NaCl exchange happen between ascending limb and this descending limb of vasa recta. एंड ऑब्वियसली सेम वे जो पानी नीचे आ रहा था, पानी निकल रहा है बाहर, दिस वाटर इज बेसिकली टू मेंटेन दिस वाटर बैलेंस, NaCl यहां से भर भर के बाहर आ गया. So that means this NaCl which is coming out maintaining high osmolarity in medulla, and finally balance of the मतलब what blood balance is maintained. Blood में दोबारा से 300 हो गया, it is already maintained, right? So now again the blood concentration become 300 maintained, it will go to the body again. So बच्चा vasa recta and ascending this loop of Henle exchanging the water NaCl in such a way that it will maintain high NaCl बाहर, NaCl more outside. इस साइड भी पूरा NaCl is maintained high outside, and some amount of urea released by collecting duct, so that solute level in medulla will be higher and 1200 osmolarity will be maintained in inner medulla. तो ये हमारा purpose है, done by NaCl and urea. So I'm pretty sure so far everything is clear to everyone, right? And बच्चा now why this has happened? ऐसा क्यों हुआ? हमने इतना ज़्यादा osmolarity क्यों बढ़ाया? So that इतना solute क्यों बढ़ा दिया? Why we have increased so much solute in medulla? So that water that is coming in the 200 milliosmolarity water है in this particular duct. So as the water coming down, तो हमारा पर्पस था टू मेंटेन कॉन्सेंट्रेशन ग्रेडियंट. कॉन्सेंट्रेशन ग्रेडियंट मीन्स ऑब्वियसली आवर पर्पस वॉज टू मेंटेन कॉन्सेंट्रेशन ग्रेडियंट. कॉन्सेंट्रेशन ग्रेडियंट मीन्स डिफरेंसेस, right? So what we have done, we have basically created differences, concentration difference को maintain कर दिया. अब इससे क्या होगा? You know it very well that obviously collecting duct receiving the filtrate, और उस filtrate में, उसके अंदर filtrate में 200 milliosmolarity is there, which is very low, and medulla have 1200 milliosmolarity, which is very high. So we know that if the concentration gradient is more water will come out to balance it, and hence what will happen because of that, जब ज़्यादा पानी बाहर आएगा just to maintain high osmolarity outside, क्योंकि बाहर ज़्यादा solute है, उसको बैलेंस करने के लिए water will come out. So because of this concentration difference की वजह से 200 एक 1200, तो जल्दी जल्दी water will come out from collecting duct, and obviously concentrated urine will be formed. अब urine जो निकलेगी, it will be having less water loss, right? So urine will be so concentrated. कितनी concentrated? 1200 milliosmolarity per liter. इतना concentrated means huge solute, less water. Initially filtrate were 300 milliosmolarity, but now the urine having the 1200 milliosmolarity. 4 times concentrated urine is formed. So why this concentration gradient is maintained between medulla and filtrate? तो बच्चा ये इसलिए किया जिससे कि huge amount of water can be reabsorbed back in the body fluid, and concentrated urine, highly concentrated urine can be formed. How much concentrated? 4 times. So highly concentrated urine is released. So bacha, is it clear to everyone? That's all, right? So how much concentrated? So initial filtrate से 4 times. Initial filtrate से 4 times concentrated, because initial filtrate having 300 milliosmol, but now it is having the 1200 milliosmol, that you have to remember, right? So initial filtrate का कितना था? 300 milliosmol. But now the urine become 4 times highly concentrated. You have to remember this. It become 1200 milliosmoles. तो we are releasing highly concentrated urine. था ना amazing? And बच्चा, whatever doubt you are having, you will be asking me, right? तो बच्चा, see, this is the ascending limb releasing NaCl going to the vasa recta, and then again vasa recta releasing NaCl here outside, और पानी अब इसमें जा रहा है. तो this is actually counter current mechanism, opposite flow of the fluid and the blood in the ascending limb, descending limb of vasa recta as well as loop of Henle. Right, so that's all, and we will be continuing our micturition reflex in the third part. See you all in the next class. I hope you liked the video. Please write down in the comment box for the notes and the doubts. Write down, send me there in the channel also, in the comment also, right? So have a great day. Good night. Bye bye, bacha. See you all in the next class.